Chapter 5: The Structure and Function of Large Biological Molecules. When we eat food, we're ingesting large biomolecules that will be used in chemical reactions inside your body to create new molecules and provide energy to maintain homeostasis. Sugars will need to be broken down with enzymes to create ATP. Fats will need to be recycled into new lipids for creating cell membranes. Proteins are broken down to reassemble and make new proteins. All of the necessary building blocks for making new cells and tissues comes from the raw materials going in. Some have to be processed before they can be used, and some are only made so that they can help process other molecules. So when you hear someone say that you are what you eat, know that we literally are what we eat. The molecules of life. Within all cells, small organic molecules are joined together to form larger molecules. Biological macromolecules are the large molecules necessary for life. All living things are made up of 4 classes of macromolecules: carbohydrates, lipids, proteins, nucleic acids. These large macromolecules may consist of thousands of covalently bonded atoms, some with mass greater than 100,000 daltons. Biochemists have determined the detailed structures of many macromolecules, which exhibit unique emergent properties arising from the orderly arrangement of their atoms. Why is the structure of a protein important for its function? As we learn in biology, structure determines function. Concept 5.1. Macromolecules are polymers built from monomers. 3 of the 4 classes of macromolecules, carbohydrates, proteins, and nucleic acids, form chain-like molecules called polymers. A polymer is a long molecule consisting of many similar or identical building blocks linked by covalent bonds. The repeated units are small molecules called monomers. Some of the molecules that serve as monomers have other functions of their own. The chemical mechanisms which cells use to make and break polymers are similar for all classes of macromolecules. These processes are facilitated by enzymes, which are specialized macromolecules that speed up chemical reactions in cells. Monomers are connected by covalent bonds that form through the loss of a water molecule. This reaction is called a dehydration reaction. When a bond forms between 2 monomers, each monomer contributes part of the water molecule that is lost. One monomer provides a hydroxyl group, the OH, while the other provides a hydrogen atom. Cells use energy to carry out dehydration reactions. The covalent bonds that connect monomers in a polymer are disassembled by hydrolysis, a reaction that is effectively the reverse of dehydration. In hydrolysis, bonds are broken by the addition of water molecules. A hydrogen atom is attached to one monomer, and a hydroxyl group attaches to the adjacent monomer. The process of digestion is an example of hydrolysis within the human body. We take in food as organic polymers that are too large for our cells to absorb. In the digestive tract, enzymes direct the hydrolysis of specific polymers. The resulting monomers are absorbed by the cells lining the gut and transported to the bloodstream for distribution to the body cells. The synthesis and breakdown of polymers. The cells of our body then use dehydration reactions to assemble the monomers into new and different polymers that carry out functions specific to the particular cell type. An immense variety of polymers can be built from a small number of monomers. Each cell has thousands of different kinds of macromolecules. Macromolecules vary among cells of the same individual. They vary more among unrelated individuals of a species and even more between species. The diversity comes from various combinations of the 40 to 50 common monomers and some others that rarely occur. These monomers can be connected in a great many combinations, just as the 26 letters in the alphabet are used to create a whole dictionary of words. The molecular logic of life is simple but elegant. Small molecules common to all organisms are ordered into unique macromolecules. Despite the great diversity in organic molecules, members of each of the 4 major classes of macromolecules are similar in structure and function.